Today we'll be talking about beta sheets. Okay, so beta sheets are essentially secondary structures, secondary protein structures, similar to alpha helixes. So both the alpha helix and the beta sheet are going to be secondary protein structures. Okay, so they're essentially a collection of amino acids. Okay, so secondary protein structures. I'm just going to put struct, okay? Structures. And there's a difference between the alpha helix and the protein sheet, the beta sheet. So remember, alpha helixes, they look like phone cords, right? They'll look like little ribbons that are being turned in each other. However, beta sheets essentially look like a crumpled up piece of paper. So if you remember when you were doing cyclohexanes, right, in 3D in organic chemistry, you would have those little ridges, right? So you would have something like this, right? So it kind of looks like that structure right there, right? Very similar. Okay. But the difference between alpha helix and the beta sheet is that the alpha helix only uses one polypeptide chain, okay? Let's just put pep for peptide. Okay. So for instance, you would have this little ribbon. Okay, and that is one polypeptide chain. So you just have like a chain of alanine, leucine, etc., right? Okay? And then you would have another one. And that's going to be like cytosine, isoleucine, whatever. Cool. But, well, these are individuals. So that's an alpha helix. And this is also an alpha helix. Okay? But with beta sheets, they are actually made from two or more polypeptide chains, okay? And so if you were to combine a polypeptide chain with another polypeptide chain and like kind of link it together, they're going to form a beta sheet. So beta sheets, you could say that beta sheets use two or more polypep chains, okay? Chains. Cool. And it is important for you to know that usually one strand, let's call this the A strand, and let's call this the B strand. Usually beta sheets are made from hydrophobic polypeptide chains combined with hydrophilic polypeptide chains, okay? So let's say that A is going to be hydrophilic. You know, I'm just putting that randomly. It could be hydrophobic for all I know. So this could be like hydrophobic. And this one could be hydrophilic. Okay? Well, you want to do this because it's actually going to offer kind of like stability within the beta sheet. Now, also, beta sheets, okay, beta sheets are not usually soluble in water. Like, yeah, they can be soluble in water, but not to like the greatest extent, okay? So they have a limited solubility in water. Alright, okay. And that's because they have, of course, the hydrophobic chain. Now, you don't want to have one chain that is mixed with hydrophobic or hydrophilic. So you don't want to have alanine and then serine, alanine, serine. That's not going to work because of sterics. Remember, you don't want polar opposites really next to each other. So you want essentially a negative side combined with a positive side on opposite ends of the beta sheet, okay? And of course, beta sheets are similar to alpha helixes in the way that, or helices, in the way that you read them, okay? So you read them from the positive side to the negative side. That's always going to be the case in biochemistry, okay? So in this example, you have, specifically for the A chain, you start reading it from the N terminus to the C terminal. So let's do this in a nice purple color, okay? Also, it's kind of important that you know that this one is going to be what? This one is going to be antiparallel, okay? Antiparallel. How do I know that? Well, let's call that antiparallel. Well, if you notice that we're reading from left to right, from the positive to the negative, right? Okay, well, on the other side, on chain B, you're reading it from the right side to the left side, right? So positive to negative runs the opposite way. Sometimes, if you're looking at a computer model, okay, let's say you put up a Gaussian application on the computer in lab, and you don't know where the nitrogens are, you don't know where the carbons are. There is a trick for you to know if this is a parallel or anti-parallel beta sheet. And the trick is to look at the hydrogen bonds, okay? So of course, alpha helices and beta sheets utilize hydrogen bonds to form these structures. And if you look at this right here, they are like that. They are going very straight, okay? And essentially, if you have a straight bondage between the oxygen of the carboxyl group and the hydrogen of the amino group, then it's going to be anti-parallel, okay? So this is anti-parallel. Let's put it here. So anti-par, if let's just put this, where R groups can be like some sort of random amino acid, is bonded like this. Okay, so this is anti-parallel, right? So what is parallel? Well, parallel would have to be this guy. Parallel is like this. Essentially, it's when when the oxygen is kind of bonded at an angle for the hydrogen and nitrogen. So it's kind of like, it's kind of ironic, right? You would expect this guy to be parallel because the hydrogen and the oxygen are parallel. And you would expect this one to be anti-parallel because the oxygen is at an angle to the hydrogen. But that's not the case. Think of opposites, okay? And anti-parallel is going to be the best. So this is the best. The best, okay? It's the best for beta sheets because it's kind of like, how should I say? It's because the beta sheets, whenever they're in anti-parallel, they're already kind of like twisting and turning, okay? So imagine you have a very weak ladder, okay? Like a ladder made of rope, right? And you have the little ledges in the rope so you can climb, right? Are you picturing the ladder? Kind of like this, right? So right here, this is pretty good. Let's say that the nitrogen is here and the carbon's here, and then nitrogen's here, carbon's there, right? So this is going to be an anti-parallel beta sheet, and the direction is right here. So imagine how you have like a 50-foot ladder made of rope like that. If you were to kind of grab the edges and kind of twist towards the right, that is going to kind of form well, obviously it's going to form some stress onto the beta sheet, right? And since there's since the beta sheet is already twisting by itself, like the nitrogen going upwards and the other nitrogen going downwards, there's already some stress there. So it's already used to the stress of twists, right? So if you were to add some more twists and some more stress, it could take it because it's already made with stress in it, right? And so they have a greater stability to solvents that try to twist it even more, right? But for parallel beta sheets, where the nitrogen is going this way, well, they're not used to the stress. They're used to going in one direction, right? So if you add a solvent that tries to twist it, that tries to make it go in a different direction, it's just going to collapse, right? If you take a person who has been working out in the gym for about 16 years and you tell them to bench press 200 pounds, they're going to be used to it. They're going to be doing it quite well, right? So they're going to be stronger because they're already used to the stress. However, if you tell someone who is on their first day of working out and you tell them to bench press 200 pounds, it's not going to be so good. They're not used to the stress. They're not used to that environment. Okay, so beta sheets are usually anti-parallel, okay, because it provides some stability in their structures, okay? Beta sheets are usually made from very small amino acids, okay, like alanine or glycine, right? And that just kind of offers some more stability as well. And also the R groups, if you noticed, the reason why I mentioned cyclohexane drawings in organic chemistry was because you always put your groups either equatorial or axial, right? So in the same vein of thought, the beta sheet has its R groups pointing downwards, and on the other side, the R groups are pointed upwards, right? So they're in opposite directions, okay? So that's like a anti-parallel structure, right? So here we go. So the R groups are usually pointing downwards or upwards, right? So they're not pointing towards the sides. So they have kind of like a 3D shape to them, right? And that's why you see kind of like the plated sheets right there. So the plated sheets, like the wrinkles in the piece of paper, are actually kind of like the R groups pointing upwards, okay? And the R groups pointing downwards. So you always have like an equal amount of ups and downs, and that creates the wrinkles within the beta sheets. It also offers structural support and rigidity, right? So firmness. If you didn't have that, you would just have a piece of paper. It's not good, okay? For instance, if you have an example, you could get like a flat sheet of paper, put some coins on it, and it'll eventually collapse, right? But if you were to put some wrinkles on it, and puts the same amount of coins, you can actually put more coins because the folds provide more support for the paper. So the ups and downs of the R chains provide more support to the beta sheet. So that offers a lot of strength, usually within spider webs or silk or other natural fibers that are very strong. Okay, so R groups point up and down. And also, the beta sheets are usually made from 3 to 10 amino acids. So, beta sheets. Ooh, that was a bad beta. That's, whatever. We'll just put that. Beta sheets are 3 to 10 amino acids long. Okay? So here is an example between anti-parallel beta sheets and parallel beta sheets, right? So of course, remember what I told you is that this is going to be the parallel beta sheet, which is really not favorable. So this is parallel beta sheet, and this is going to be anti-parallel. Well, yeah, I guess you could call it anti-beta sheet. Yeah, whatever, right? Okay, so how can you tell? Well, notice that these have kind of like angled bonds between the oxygen and hydrogen, okay? And also notice that over here, this is going to be a vertical bond between the oxygen and hydrogen. If you're looking at the chain structure like this, right? So you have like the letters and the elements showing. Well, you can realize that you have the nitrogen right here. So that's going to be positive. That's going to be positive, and that's going to be positive. And then over here you have the negative carbons, right? And of course we go from positive to negative, so we go over here like that. Okay, so they're all going in the same direction, left to right, so that's going to be a parallel configuration. Over here it looks like a spaghetti, right? Or it just looks like a mess, okay? Like word soup. So alphabet soup, there we go. So you just have like a bunch of letters, but you have to realize something. You have to realize that over here is a carbon, okay? So let's say that this is carbon right there. Actually, if you want to be really specific, there's a nitrogen. So that's positive. And at the very end, there's a carbon right here. Okay, so carbon is negative. And then we go to the other chain, and we see that there's a carbon right here. Okay, so therefore, there's going to be a C negative. And if we look over here, there's a nitrogen. So N plus. We go here, there is what? A nitrogen. N plus, and carbon is going to be the last one, C negative. Okay, so we go from positive to negative. So it's like that. Positive to negative goes like this. Positive to negative goes like that. So overall, you have kind of like a zigzag formation. Okay, so this is going to be antiparallel. It's favorable. It's going to have more structure and more rigidity, more support. Okay, so that's how you figure out which one is parallel and which one is anti. And remember that anti is the best beta sheet. Beta sheets also have the possibility to kind of bend on itself. Now, bends are typically associated with those weird amino acids that I told you. There's two of them, which is proline and glycine. Okay, or yes, glycine, right? I don't know why it just kind of froze up. But so proline was just kind of do like this, H2N, carbon, right? They have H, and also this guy, right? So proline and glycine, right? So glycine. Those are going to create bends and kinks in the beta sheet, okay? So they create bends. Now, bends can actually kind of change the direction of which the beta sheet is read, okay? So instead of being left to right, positive to negative, it could kind of change. So it could be like a positive to negative on the opposite direction, right? So instead of having this, you will have this now, which kind of changes how the beta sheet is read and how it reacts within the body or in different reactions, okay? It can form a different structure, it can do a different thing. Okay, so these do not typically happen too much, right? Because you don't really want to use proline and glycine in the first place, right? So these are irregular. Okay, these are irregular. Sometimes they differ in length, okay? So sometimes they can kind of range from about 6 to 16 amino acids, I would say. So like one of the longest chains could be 6 to 16 amino acids, right? That can do that, or about 10 angstrom long, angstroms, right? So of course you go from a C negative, you're being read right there, and this is an N positive, and now they're kind of like changing how it's read, okay? Now the bends can actually connect anti-parallel beta sheets. So bends can connect anti-parallel beta sheets. Okay, and of course that can actually, change how the beta sheet is read. Okay, how you read it. So the specific name right here would be called a beta bend or a hairpin twist or bend. Now the hairpin bend comes from a bobby pin, so sometimes people with long hair put bobby pins in their hair. So if you take one and you look at it, you can imagine that's what's happening within the amino acids in the beta sheet, right? So you have this little weird thing right here, right? And these guys are actually connecting in on itself, right? So you have kind of like this, right? So the reason why it happens with antiparallel is because they're already horizontal to each other, so it's easier to kind of connect in on itself. However, if you were to have the same thing, the same bobby pin, right, and it's antiparallel, it would be really weird to do it, right? So it would be kind of awkward to try to connect on itself at an angle. That's not going to happen, okay? So it's most likely to occur on anti-parallel beta sheets. Not that it can't occur on parallel beta sheets, but it's going to happen most likely on anti-parallel beta sheets when you use proline or glycine, okay? So there you go, yeah. And now we will be talking about motifs or motifs, right? So essentially what they are, they're just really nice pictures, okay? That's all they are. So sometimes in your book, if you're reading it, you'll see these beautiful, elegant pictures of structures. For instance, the alpha helix has a nice ribbon-like structure. The beta sheet has these kind of like, they look like little macaronis, okay? So they have these nice structures for different proteins, okay? And that's just computer generated. So motifs are just a collection of patterns that represent the protein structures, okay? So just, 3D images or patterns of protein structures. More specifically, secondary protein structures. Okay, so everything we're doing right now is for secondary protein structures, and essentially, they're just they're arranged in like the most stable geometries. Okay, and if you want to get into the specifics, they're often called ribbon diagrams. Okay, and so alpha helixes are going to have these cylinders or coils. They look like ribbons, for what I've been saying since day one. They look like ribbons, right? So these have cylinders and coils. And coils. Okay, those have cylinders and coils. And over here, you're going to have the beta sheets. Now, what these are called individually are called beta strands, okay? So each one individually is called a beta strand. And beta strands actually show direction. So this one you can't tell because normally ribbon diagrams don't show the amino acids. They don't show the N terminus to C terminus. They just show the shape, right? But over here, for beta strands, they actually do show the N terminus and C terminus, okay? And you're gonna have to use some brain power to figure out the direction. So over here, you can say that this one, since we're going from left to right, this would have to be the N terminus, right there, positive, and this would have to be the C terminus, okay? Because we go from positive to negative. Over here would have the opposite, so this is N plus, and this is C negative, and then this would have to be N plus and C negative. Now, I kind of blew up the image of the second portion over here. So right here, you would have one individual polypeptide chain, remember? Because beta sheets actually use two polypeptide chains. One goes in one direction, the other one goes in another direction, or in the same direction, depending on the beta sheet. So let's just say that we have the most simplest example, right? Alanine, okay? So we have alanine, 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 right? So that's a good example. And then over here we can have tyrosine or something, right? Now let's just use a serine. Because serine's polar and this is nonpolar, I just want to make it more realistic. Okay, so over there, we know that this is going from positive to negative, so we can say that this portion is going to have its N terminus right here, and its C terminus over there. And over here we have the thing going from right to left. So we go from C terminus, or actually N terminus, to C terminus. And we can tell that this is going to be what? This is anti-parallel. Okay, so anti-parallel, which is ideal, right? And over here you would have the same thing, except it would be in a same direction. okay? So we would have the N terminus right here, and the C terminus, and then the N terminus, and the C terminus, okay? Over there, you would notice that right here, there is a turn, right? So turns are very prominent within antiparallel configurations. So this is kind of like the, it's going to connect on each other. That's what I'm trying to say. So this one is kind of like a turn that connects. Okay, and you can see right here that turns are more favorable within the anti-parallel configuration. And again, if you don't see the elements, you can just tell, hey, this guy is staggered, right? And this guy is at an angle as well. So most likely, these are going to be anti-parallel. Now we will be looking at the combination of motifs or motifs within protein structures. So essentially you can combine both alpha helices, beta sheets, and what else? Turns, right? So quick reminder that beta sheets are made up of beta strands, okay? So that's just a reminder. Right here, you can tell that, what are these? Are these beta sheets? No. You have a ribbon-like structure. You have kind of like a coil. So this is going to be what? This is going to be a alpha helix, okay? So we have alpha helix for the first part. Over here, we're going to have another portion that's a turn. So we're going to put alpha, and then we're going to put a T for turn, okay? And then we have another alpha helix. So that's going to be like this, okay? Excuse me. So that's an alpha helix, turn, alpha helix motif. And specifically, if you want to get into it, this is a calcium binding motif. Okay, so this binds calcium. Okay, and it's like connected to 3 aspartamines or whatever. So that's not really important. Now we can actually judge the structure if it's parallel or anti-parallel. So we can just do that by doing a positive. So N positive goes down, so it goes like this. Okay. And of course that actually goes to the right. So this is always right-handed for proteins. Okay. And then it follows a turn, and it goes again like this. Right. So that's a C negative. So of course this is going to be going over here, goes there. So this is anti-parallel. Okay. Now over here, you also have the turn, but instead you're not working with coils, right? You're not working with coils. You're working with beta sheets. Okay. Working with beta sheets. So we just do the same thing. We put beta, turn, and then another beta. Okay. So that's a beta, turn, beta motif. And of course you can tell the parallel or anti-parallel. So N positive, it goes down, and C negative is going to go up. So this is anti-parallel. Over here it's a bit more complicated. Okay, it's a bit more complicated, but we can do it. So we have a beta sheet. Okay, so we have beta, turn, and then beta, turn, beta, turn. So let's see, 1, 2, 3, and then we have 1, 2, 3 turns, and another beta. And we go from N positive, it's going to be C negative. We go down here, that's going to be N positive, C negative, N positive, C negative, and N positive, C negative. Okay, so we go up, down, up, and down, right? So looking at that, that is going to be anti-parallel. Very, very nice, okay? Right there. And finally, we have this guy, okay? We have this guy. So we have N positive and C negative. Then we go to N positive, C negative. So we know that this is going to be a parallel structure, and that's that good. Also, this right here, would have to be an alpha helix. Okay. So the thing is, when you're judging alpha helices compared to beta sheets, you want to kind of keep them in their own domain, meaning that, okay, well, technically, it's N positive to, C negative, so it's always going to be antiparallel. No. You only do antiparallel or parallel for beta sheets, okay? You do not incorporate the idea of an alpha helix. So you only care about beta sheets when you're talking about parallel or antiparallel. So if we're just looking at the beta sheets, they are going upwards, okay? And this one is going to go upwards as well. So just looking at the beta sheets, we can tell that the beta sheets are going to be a parallel configuration. So overall, the system, or the structure, I should say, is going to be parallel, which is not ideal, okay? So if you write this out, this would be a beta, alpha, beta, motif, or motif, whatever. So there you go. That's how you kind of designate the direction and the names for different protein structures, okay? So again, you can combine protein structures and turns. We're going to talk about fibrous proteins, like very quickly, right? So fibrous proteins are just kind of like a combination of, should I say, of just of amino acids, I guess. But it's kind of weird because, for instance, let's talk about collagen, okay? So for fibrous proteins, the main one that you need to know is a collagen, okay? Collagen, because that is going to make up a large portion of the bones, skin, and tendons. Okay, there you go. So the weird thing about it is that collagen has a lot, and I mean a lot of protein. Okay, so has a bunch of protein. Okay. So what does that tell us? It tells us that if you have an excessive amount of proline, you're not going to be able to form alpha helices or beta sheets because those are too big and bulky, okay? So collagen can't form alpha helixes or helices or beta sheets. Okay, so that's really weird. But also notice that fibrous proteins, like collagen, have a triple twist, right? So they kind of like form a helix shape, but it's not an alpha helix, okay? It's just like a helix, okay? So it forms a helix, but it is not the alpha helix, okay? So they have like a helix shape that is big and bulky, and the more turns it has, the more strength it has, okay? So you have a bunch of turns, and that makes it really, really strong. And so fibrous proteins, like collagen, they actually have very high strength. So high, if you want it to be like an engineer, you can say high tensile strength. I can't spell, can I? So tensile strength is kind of like the basic example that I can give you is like if you get a strand of hair and you're able to hang 10 pounds of weight on it from one single strand of hair, you can say that it has a high tensile strength. So it's kind of like the capacity to carry a large amount of weight within a certain length and diameter, okay? So high tensile strength is very common for fibrous proteins. Fibrous proteins can't usually make alpha helixes or beta sheets, specifically for collagen. So collagen is one of the main fibrous proteins in the secondary structure, okay? So to recap, collagen cannot form alpha helixes or beta sheets. It has a helix shape and it has a high tensile strength. So now, whenever you bring everything that we talked about, like literally everything that I talked about with proteins, alpha helices, beta sheets, turns, amino acids, pigs even, they form to make the tertiary structure, okay? So the tertiary structure, is the global arrangement for all the atoms that we talked about. So the global arrangement of all atoms. Atons, what? No. So atoms, actually. So atoms. Okay, so essentially, from like the most basic monomer that makes up the polymer, that makes up the amino acid, that makes up the alpha helix, that joins with the beta sheet, that combines together to make the tertiary structure, that is the global arrangement of all the atoms, okay? And so essentially you're going to have regions of this structure where the beta sheets interact with each other, where the alpha helices interact with each other, and they form different complex systems, okay? So you can say that, like the joining of secondary structures. So when secondary structures join, they make tertiary structures. Okay. Now you can have amino acids interacting with each other. You can also have prosthetic groups interacting with each other. Prosthetic. So a prosthetic group is essentially a protein structure that has like a small organic molecule next to it, okay? Or it can have a metal ion next to it. So for instance, you can have like a protein group with like a potassium ion next to it, and that would be called a prosthetic group. It can also be a metal as well. So like copper next to the protein would be a prosthetic group, okay? So a major force, a major joining force for the tertiary structure would have to be the hydrophobic amino acids or compounds found in the core of the tertiary structure. Now, the reason for that goes back to one of the old videos I made talking about the hydrophobic effect, right? So the hydrophobic effect is actually going to play a key role in the formation of proteins, okay? So essentially, let's say that everything around here on the outside world is water, okay? Is water. So you're going to have your hydrophobic amino acids like alanine, or leucine or isoleucine being scared. They don't want to interact with the water. If they do interact with the water, they're going to drive up the amount of entropy and the amount of energy needed to maintain that form. So they don't want to interact with water. They're not hydrophilic. They want to stay hidden inside the structure, right? And so they're going to push the polar, or you should say hydrophilic amino acids towards the outside. So for instance, alanine is going to push out serine, and serine is going to be interacting with the water. Now, it is that driving force that pushes the hydrophobic amino acids inside and maintains that structure for the protein complex, okay? So again, the hydrophobic force dictates or makes the protein structure. It makes the tertiary structure, if you want to say, okay? So that is always a good example to go back to. So hydrophobic force or effect, you can say, is just when the hydrophobic amino acids don't want to interact with the water, so they kind of convert the structure overall to do that. So for instance, you can have a beta sheet made entirely of hydrophobic compounds, okay? Well, is that beta sheet going to be outside? No, it's going to be inside the core. And if you keep doing that, if you keep putting beta sheets that are hydrophilic outside and the beta sheets that are hydrophobic inside, when they combine, they're going to make a different structure, and they're going to be held in place because that is the most favorable structure to do energy-wise. Okay, so hopefully that explains why hydrophobic forces, or the joining force, like a major joining force, where you're talking about tertiary structures. So another example, to kind of reiterate the hydrophobic effect, the proteins always want to have the lowest amount of energy, okay? Because if they have too much energy to make something, it's not going to be efficient, right? So, for instance, some people prefer to go and to Jack in the Box or McDonald's or whatever, because it's more time efficient, they get more calories in or whatever, and it takes more energy to cook the food. So in that regard, people would rather waste the least amount of energy. So proteins, you're kind of lazy. They don't want to waste more energy to do what they need to do, right? So whenever you're unfolded, so you have like a alanine, tyrosine or whatever linked up together, just on like a straight line, it's going to have more energy, okay? Because it's going to be interacting with the surrounding environment. For instance, let's say water. And if you have a hydrophobic protein structure, right, or a polypeptide, then it's going to be interacting with water. It's not that good, So now you're going to introduce kind of like protein folding, okay? So this is protein folding. And essentially, protein folding is kind of like making the protein, which is, let's say, a mile long, into a compacted state that is about an inch long or something, right? So as you go downwards, you're going to have a decrease in energy. So decreased energy, it's favorable, which is favorable. Okay. So as you do that, you're going to slightly fold and fold. And when you start folding the protein structures, you can introduce alpha helices, beta sheets, and whatever, okay? So there you go. So whenever you successfully fold it, you will actually decrease the energy and make it more favorable. So on another example, we have the same structure, right? So we have the highest energy, which is not good, and then we have the lowest energy. So you can see that when you start folding certain polypeptides, you can introduce new forms, for instance, alpha helices and beta sheets, right? So now you're going to go into like the motif. So whenever you do that, as you start folding and folding, let's say that the beta sheet is going to be hydrophobic because it's made of hydrophobic amino acids. Okay, let's just say that, let's say. And the alpha helices in that example would actually be hydrophilic, so they want to be on the outside. So they know this, and they say, the beta sheet is like, okay, look, before we start folding, I want to be on the inside because I don't want to be interacting with water. That's going to waste too much energy. And the alpha helices roll their eyes and they're like, whatever, let's just do it, okay? It's a 9-to-5 job. I get paid like $7 an hour. Let's just go on with it, right? It's not even minimum wage. But they start folding each other, and the beta sheet is being protected by the alpha helix in this example. So whenever it reaches its final stage, which is going to be like the tertiary structure, right? Because right now you're just introducing kind of like the secondary structure, and this is the primary structure, right? So tertiary structure is kind of like the end product for one complex, and we're going to talk about quaternary structures later, okay? As a hint, quaternary structures are just a collection of tertiary structures. So yeah. So now the beta sheets, which are hydrophobic in this example, are being protected by the alpha helices, which are hydrophilic. And that is kind of pushed to by the hydrophobic effect. Okay, so that's going to be the reason why they stick together, the reason why they stay that way, and the reason why they formed that structure like that in the first place. Okay. And before I forget, there is something called a molten globule. So this is a molten globule. Okay, and essentially it's almost completely compact. Almost. So it's a compact protein structure that is almost the active native species. Okay, so it's going to have a large portion of its amino acids interacting with the water, and the problem is that molten globules are typically hydrophobic. Okay, so that's not a really good state. it's kind of like, oh, we're getting there, we're getting to like the perfect form, but still we're not that good. So molten globules are kind of like the next step, the step before the native species or the biologically active protein. Now you get to the point in life where you combine the tertiary structures into a different structure. So let's call the tertiary structures complexes, okay? So these complexes combine and they make a quaternary structure. Okay, so now we're talking about quaternary structures. This is the last structure, okay? Right. So these quaternary structures eventually will become a different part of an organ or a part of a system or something. But essentially they're made from tiny pieces or tiny individuals of tertiary structures, okay? So you can see how complex it gets, right? So depending on how many tertiary structures there are, a quaternary structure can be called a dimer, trimer, quatmer, socmer, hexamer. So there's like different names for them, okay? So whenever you have a quaternary structure that is made from from multiple tertiary structures, we call them oligomers, okay? And oligomers are essentially, it's just a structure made from different tertiary structures, okay? If a quaternary structure is made from identical complexes, right, then you would call them hetero, or actually homo oligomers, sorry. So identical complexes is going to be called a homo oligomer. Okay? Now if this is made from different complexes, then we're going to call that a hetero oligomer. So that's going to be called a hetero. Okay? So what is an example of a hetero oligomer? Well, if you look over here, this is actually hemoglobin. Okay, so hemoglobin is a very important transport protein, especially within the carbonic acid bicarbonate buffer system. Okay, so this is the hemoglobin protein. So it's a quaternary structure that's made from tertiary structures, which themselves are made from secondary structures, and so on and so forth. Right, so this hemoglobin protein is going to be called what? This is called a hetero tetramer. Why? Well, it's hetero because notice that it has two beta sheets, and also has two alpha chains, okay? Or alpha helices, sorry. So because it has two beta sheets and two alpha helices, they are going to add up to 4. So 2 plus 2 is 4. That means tetra, right? And oligomer, the MER comes from oligomer. And because they're made of different complexes, alpha helix is different from a beta sheet, we're going to call that hetero. So together we put hetero tetramer, okay? So hetero tetramer. So there are some structures that are stabilized by non-covalent forces, okay? So by non-covalent forces, I mean by electrostatic attraction, okay? So that's when, oxygen is connected to the hydrogen. Now that can also be called what? Hydrogen bonding, right? Actually, sorry. So, so electrostatic attraction is just when the carboxyl group, for instance, in a protein, is bonded to the amino group in a protein. Okay, and hydrogen bonding, self-explanatory, is when the oxygen is connected to the hydrogen. Okay, now another force would be the metal-ion coordination. So, for instance, that would introduce prosthetics into the protein fold, right? So you have a protein that has a metal ion next to it, right? So that would be a metal-ion coordination. An example would be like copper in the body. Okay, Now you got the disulfide bond. So the disulfide bond, I think, is pretty important to realize that this is actually a covalent bond. So you're probably wondering, hey, I thought you said this is non-covalent. Well, the exception to that rule would have to be the disulfide bond, which you find in cysteine. So you find this guy in cysteine, right? Cysteine, right here, okay? And that's often found in hair. That's why you can break those bonds to like make a crazy hairdo, right? So that's the exception. This is actually a covalent bond, okay? Then you got your hydrophobic interactions, which is actually the main force used to make protein structures the way they are, okay? You got the helical structure, which provides support and kind of like rigidity to the alpha helix. And then your sheet structures for the beta sheets, obviously, okay? So once you combine protein structures, it's important to know that they are not set in stone, okay? They're not completely rigid. This isn't going to be permanent. So protein structures can change. So you remember when we were talking about protein folding, of how you always try to go to the lowest amount of energy? Well, that can actually be reversible. So depending on what you're doing in the lab, you can actually treat, let's say for instance, hair with a reagent. Okay, so if you want to change the disulfide bond, you can put something in your hair that will actually break the bonds and weaken the structure. So you have this unfolding of proteins, okay? And then you can kind of reinstate the bonds in a new position using hydrogen peroxide, right? So you can actually reverse the process and then convert it back. So essentially, this is called denaturation or denature. So denaturing. Right, so whenever a protein denatures, it kind of loses its shape and it has more energy, and that's not really good. So usually you can convert denatured proteins back into their structures. However, the exception would have to be heat. So heat denatured proteins can't fold back, and that's really bad. Okay, so whenever your body gets too hot or proteins are exposed to increased temperatures, they lose the ability to bind with enzymes and they cannot fold back. Okay, so that's really, really bad. Okay, so always try to avoid that. And finally, before I wrap up, why are we studying protein structures? Like, who cares, man? Well, the reason why we study protein structures is because a lot of diseases actually stem from protein structures. For instance, cystic fibrosis is a issue with the primary structure, amino acids. Okay, so how the amino acids bond, how they replicate, how they are in order, that is changed in cystic fibrosis. And for those of you who don't know what cystic fibrosis is, it's essentially when the body, the endocrine system, like pours out mucus from the skin, okay? So you're excreting like a thin, oily film from your skin. And it's just really bad, right? For males, for guys, it causes sterility problems, okay? So you're not able to have children. And there's about 1,000 cases per year in America because of cystic fibrosis. And that's just the changes in the amino acid chain, like the polypeptide chain in the primary structure. So that's like really bad, okay? Another example would have to be sickle cell anemia, which is an issue within like the tertiary structure of the hemoglobin or whatnot. They're not functioning properly, they're not forming properly. You can get low iron deficiency. You can faint, you are most likely tired a lot of the time, and that stems from a mutation within the hemoglobin. So they're not forming protein structures like they should, okay? like the protein structures that are made are kind of wonky, and that's not good. So whenever you're debating, why am I taking this course? Why am I taking biochemistry? You're trying to understand the protein structures that make up diseases, how it changes, and to see if you can make something that combats sickle cell anemia. So maybe you might be the person who makes medication that completely stops sickle cell anemia, So, hey, that's why we're taking this course. And obviously, I hope this video helped you. I hope that you have an appreciation for your course load or something. And I hope that you have a great day. So thank you so much for spending your time with me. And I hope you do well in your exams. Remember that I will always love you. So take care, and thank you.